Hi friend, welcome to PLANTI.com. Today I am going to demonstrate titration of amino acid in biochemistry. Titration. What is titration? Titration is used to determine the concentration of an acid or base. How it is determined? Adding a sample A having known concentration but unknown volume and having a sample B known volume but unknown concentration. Suppose here present HCl, that's known volume and unknown concentration. And here present KOH, that's known concentration but unknown volume. We want to know its concentration. What is its concentration? We don't know and we want to know its concentration. That's why we slowly add KOH here at KOH with known concentration, suppose 0.1 mole concentration, and find the equivalency point. Here we used a indicator of pH change, color changes indicator that determine the pH equivalency point. Here red color. When pH reached at 7, the color will be white, and then we calculate the titration point. Amino acid. Diprotic amino acid, two types, diprotic and triprotic, which have two groups having the nature proton acceptor or donor, and which have three groups having the nature proton acceptor or donor. Here three groups are playing role in proton donor or acceptor, are here only two group are playing role proton acceptor or donor, not R group playing a role. Here diprotic amino acid, the example glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, tryptophan, etc. And triprotic, it's you have to remember. And here aspartic acid, glutamic acid, cysteine, tyrosine, histidine, arginine and lysine. Here we know that pH is varies 1 by H plus is equal to minus log H plus. Here when pH is high, then H plus will low. Here suppose COOH, we know it proton donor, it donate a proton and it becomes COO minus, and NH3 plus when donate a proton it become NH2. This condition is deprotonated condition. So when pH is low and pH is low, H plus will high. In that case, COO minus when gain proton, accept a proton, then it become COOH and NH2 when gain proton it becomes NH3 plus. This condition is protonated condition. Here pH is when low, H plus will high. Here also POH when low, OH will high. Amino acid, I previously said that which have two group having the nature proton acceptor or donor. Here I describe that suppose glycine COOH and NH2 H and H at pH condition when low, suppose 3. So here pH 3 means it is low pH, then we have high proton. Here proton concentration is high. So which group at first accept proton? We know that amino group is a highly proton acceptor and COOH is a highly proton donor. So when proton concentration is high, then NH2 group at first accept proton and it become NH3 plus. Clear? And then when it is pH become high, intermediate, pH suppose 6, then COOH, here NH3 plus, here also present NH3 plus HH. This is glycine structure. Here in this case when pH 6, then proton gradient will become low. Proton gradient here in high level proton gradient because pH low, proton high. Here pH some high, so proton gradient will some low, and then it's time to donate proton. Which donate proton? We know COOH is a high efficiency to donate proton. So COOH is here donate, so it becomes COO minus. And here in this case pH when suppose 9, in this condition NH3 plus COO minus, here H. Here also pH 9, that means H plus concentration will low. pH high means H plus low. In that case, which proton pH low means H plus low, which donate proton here NH3 plus, here it this time donate a proton because here H have no role donating proton. This H have no role donating proton, so here donate proton and it become NH2. Clear? Here I say that clearly. C here COOH, here NH3 plus, this is H and this is H, here NH3 plus, this is COO minus, this is H and H, and in that case NH2 C C double O minus H H. So in every case you see that nowhere a structure that NH2 C C double O H H nowhere present, but we always read that it's amino acid structure, but never it can possible because any pH condition it will be charging condition. Here positive charge, here neutral in charge, but both are charged. It is positive charged, it is negative charged, and it is negative charged. This is the clear concept of diprotic amino acid. Titration curve of glycine. Here it is the experiment data. Here glycine at pK1 2.38. Then what I say that it is pH low and proton condition, proton is high, and it is pi condition. Here pH is intermediate and proton will be intermediate, and pK2 here pH is high but proton is low concentration. So here how calculate it is pi 9.5.97. It is the actual use of pi calculation by titration. Titration means the pi calculation. What is the pi? Pi value. What is pi? Here calculate. How I calculate? pK1 2.34 and pK2 9.60. Here we calculate it and 11.49. This is by 2. Why 2? Because it is pK1, it is pK2. We have a formula pK1 plus pK2 by 2. Here pK1 it is and pK2 it is plus the result 11.94 by 2. Got it? You calculate it, it will be result 5.97. Got it? Some other example. Suppose pK1 here 3, pK2 here 9. What will be the pi value? What I say? pK1 plus pK2 by 2. Here pK1 3 and pK2 9 by 2 is equal to 12 by 2 to 6. So pi value will be 6. Clear? Amino acid. Here I previously say that 3 group which can accept or donate proton. 3 group. Why? Because suppose I say here told that D, E, H, R, K and C, Y. These are diprotic amino acid. Aspartic acid, glutamic acid, histidine, lysine, arginine, lysine, cysteine and tyrosine. Here suppose I accept a example aspartic acid. Here aspartic acid. So C double O H here NH3 C H C double O H here C double O H, here H, and here CH2 C double O H. This is the structure of aspartic acid. Here I wrongly write that NH2. So when pH is low, suppose we here 3, put it 3, and then proton will be high, proton condition will high, H plus proton will high. So here first accept a proton NH2 and it become NH3 plus. Then at when I pH concentration will high, pH become 5, then NH3 and pH 5, then proton will low, pH high, pH low, but proton is intermediate, not high, reach high, but here C double OH, here also CH2 C double OH, and here H, here NH3 plus, but here C double OH, it is the high proton donor, and it never want to accept proton, it always want to donate proton. So C double OH donate proton, its nature it donate proton, and it becomes C double O minus. Then here NH3 plus C double O minus H, and here CH2 C double OH. When pH condition will reach at pH 7, then what will happen? H plus concentration, pH high, H plus concentration will be low, and here also this R group COH, this is R group, it's doing nature donating proton, this COOH doing nature donating proton, and it becomes COO minus. So at the result, at pH 7, NH3 COO minus, CH2 COO minus, and here H at pH 7. When I also reach the pH 11, when pH 11, NH3 plus COO minus, CH2 COO minus, and H. When pH I reach it pH 11, so then pH high means proton will low, and then donate, here no group present for donating proton, but it need to donate proton, then NH3 donate proton, it is accepted but it is bounded to donate proton, so it is donate proton. NH3 become NH2 and COO minus, here H CH2 COO minus, it is the whole condition. So at the neat result, NH3 CH COO H, here H, here CH2 COO H, NH3 plus here at pH condition 3, when pH condition will high, NH3 COO minus H, CH2, COOH. It is pH condition 5. When NH3, COO minus, H, CH2, COO minus. It is pH condition 7. And when NH3 become NH2, COO minus, CH2, COO minus, and H. It is pH condition 11. It is the whole concept of diprotic amino acid. Okay. Then PI calculation of diprotic amino acid. How calculate diprotic amino acid? Because in diprotic condition there are pK1 and pK2, and I divided by 2 and result PI. I get PI. But here pK1 also present, pK2 present, pKr present. So how I get PI? PI value how I get? Suppose pK1 3, pK2 6, pKr 9. No, pKr 6 and pK, because here pKr in immediate condition. That's all. Because in the last condition we say that we previously get an example pH, we take it what 3, pH 5, pH 7 and pH 11. In that case 3 step, it is called pK1, it is pKr and it is pK2. So pK1 is, suppose pK1 is, I need here 3, pKr here 6 because it is second condition, and pK2 is 9. So what is PI value? What is PI value? Here I see that 2 are 3 and 6. This is the less than 7. So I see that in condition in 3 in case 2 are in less than 7. So I get it 3 plus 6 by 2 and 9 by 2, 4.5 PI value. But if it is also, suppose I get a point, suppose pK1 is equal to 5, pKr is equal to 9 and pK2 equal to 11. So here PI value, what is PI value? Then here I see that you compare it 7. If 2 are higher than 7, you calculate in this. If 2 are lower than 7, so you calculate this. But here higher than 7, 2 condition 9 and 11. So I put it 9 plus 11 by 7 by 2. 11 plus 9, 20 by 2 equal to 10. PI value here 10. Got it? Thank you for watching my class.